Okay, so here we are today looking at anaerobic respiration, and we are going to look at the anaerobic respiration of yeast. So here we have some what they call fast action dried yeast from Tesco's or Sainsbury's or any other well-known brand. So yeast is a microorganism. It's used in many things to make food, beer, wine, and what we're looking at is how yeast does anaerobic respiration, so that means without oxygen, and it produces ethanol and carbon dioxide. So we're going to do a little test today. We've got 4 test tubes here. We're going to put yeast in each of these, some water in each of these, and some sugar, so it's got something to have a little munch on. We are then going to see how it produces carbon dioxide, or we're going to prove that it produces carbon dioxide, so it should be very bubbly, giving off bubbles of carbon dioxide. We call that effervescing, so anyone in my class knows the word effervesce, that means bubbles. And we're going to try it in different temperatures to see which temperature the yeast likes. Does it like hot? Does it like cold? So what we're going to do first of all is we've got some yeast here. So this is straight from our packets. So if you were making bread at home, you would use yeast to actually do this. So we're going to put a teaspoon. Actually, I'm going to use the big spatula, might be easier. So we've got a spatula here, and we are going to try and keep it the same, keep our controls the same for each one. We're going to put 2 of these in each boiling tube. A little bit of spillage here, but that's okay. And then to that, we are then going to add some sugar. So just like us, a microorganism of any kind does require some glucose. So this is its dinner, breakfast if you will. So a scoop of sugar in each as well. And then we're going to add some water to this. So we are now going to add some water to each boiling tube. Ideally, to keep the controls the same, you'd measure this out exactly. So I'm just going to try and keep it to the level of the test tube rack. Now this process where yeast produces carbon dioxide and ethanol, remember ethanol is one of our alcohols, we talked about that in our alcohol lesson in week 7 I think, or week 2 lesson 7, and this process is called fermentation. So this is the fermentation by a microorganism called yeast to make carbon dioxide and water. So we give that little swirl around, mix it up. Again, we're going to do the same to every single test tube to try and keep our controls the same. I'm actually going to just give that a little stir just to make sure that sugar is dissolving. So it's just slightly warm water just to help the sugar dissolve. I'm then in a minute going to put a balloon on top of each of these to capture the gases. So can you remember what gas we're trying to capture? Have a little think. If you said carbon dioxide, you are correct. Does anyone know the symbols for carbon dioxide? If you said CO2, you'd be correct. So, they're all nicely mixed now. And now I've got some balloons, just normal party balloons. And we're going to put one on each of these.
These are from all the parties that got cancelled due to social distancing. We've got loads of balloons. We've got party stuff here.
Now the yeast are going to have their own party.
Yeah, having much more fun.
So ideally we'd have lots of these at each temperature, so we've got a fairer test and give us more accurate results. So these will start to ferment, they will start to produce carbon dioxide and ethanol. So now what we're going to do, we're going to take these and put them in different water baths of different temperatures. This one here is our hottest. This is at 90 degrees, so almost boiling. In we go. This this one is at 40 degrees.
Very
So
bright,
this
guys.
is,
Sorry, but that's 40.
This is the sort of temperature that our bodies like. We like to be around about 37 degrees. And this one here is at 20 degrees. And I'm also going to go and find some ice. Actually, I'm going to put this one in the fridge to see what effects really cold have. So most fridges are around about 5 degrees Celsius. So I'll put this in the fridge and see the effect of this one. So we'll come back to these later and see if any gases have been produced.
Lovely, miss. So year 8, what I'd just like you to do is make a little prediction to yourselves. Just to recap, we're going to have test tube 1 in the fridge. We've got test tube 2 at 20 degrees. Test tube 3 is at 40 and test tube 4 at the back there is at 90 degrees. Which one do you think is going to produce the most carbon dioxide? Which one do you think the yeast is going to be the happiest? Check back with you in a minute.
3 hours later.
So here we are, this is now about 3 hours later. So this is our hottest, this is our 90 degrees. As you can see, not much going on here. So let's put that in there and we'll be able to compare them all together. This is our 40 degrees. Definitely some gas has been produced there. Here we have our 20 degrees, so quite cool this one. Again, tiny bit, but not much going on there. And then finally, we have our fridge version. So if we put these together, it is very clear to see which temperature our microorganism, our yeast, prefers. So it's a bit like the 3 bears. Oh, not too hot. Oh, not too cold. Not too cold either, but it likes it at this temperature. This temperature is 40 degrees. Our body temperature is around about 37 or 37.5 degrees Celsius, and microorganisms also in general prefer this temperature as well. At this temperature, fermentation is at its most active. It is producing lots and lots of carbon dioxide. We can see the carbon dioxide bubbles here, and producing ethanol as well. So obviously if we left this all day, this is actually quite firm, and we can compare, obviously no gas at all really in here, very few bubbles going on at all. More bubbles in this one. This is our 20 degrees, and then in our fridge version there were some bubbling going on there, but actually very little has gone into our balloons. So our most successful was our 40 degrees, which is the optimum temperature for fermentation of a yeast.